Okay, so we're ready to begin the 16S experiment with preparation of the DNA sample. In this case, we happen to be using a Norgen kit. There's probably many ways that you can prepare your DNA sample of choice. At the end of the day, all that matters is that you come out with DNA that is representative of the sample that you're looking for and that it's clean enough to do PCR. So that is probably a wide range of options. We happen to be using this Norgen kit. One of the kits, and everything will be documented in the website. You can get the kit for the sterile swab and also preservative. The preservative makes sure that the DNA sample remains integral and also protects anyone from whatever microbes you might have picked up there. So it should neutralize any microbes. And then we're using the reagents from the microbiome extraction kit. If we were doing this in a classroom setting, I would aliquot these so that individual students or teams would have their own portion. And we've done this several times and it works pretty well. I'm just doing a demo, trying to pull out any tips or tricks or things that might be helpful to look out for as you prepare if you happen to be using this kit. So I haven't done any aliquots and keeping it that way. I'm wearing gloves. The samples and the amounts of things that we're working with aren't anything that's really risky, but really I'm a little bit more concerned about protecting the sample. I don't need to wear gloves for many portions of the things that I'm doing. So it's a little bit of make your decision based upon what you are doing and what is safe and what the requirements are. But the gloves in this case are more to protect the sample from me than me from the sample or the work. So I have 6 samples that I'm going to prepare today. Some of these are actually sent in by students. They're in the tube, and each one of them has a specific label that's on them. And the swab that is used to collect these, as demonstrated with the chute collection, the swab is inside. These can stay pretty much on the shelf. I don't know what the exact length of time is, but a pretty long time. These are shipped through the mail, and I think it's worked so far for us. So I'm going to begin the protocol. I'm going to go through the whole thing. There are times when I might, I don't know if I fast forward or just edit it out, but I'm just going to try to stick to the highlights enough so that anybody watching this might have an advantage in doing this themselves. So the very first thing that I need to do is to get the appropriate lysis solution. And in this case, according to my protocol, I need lysis additive A. Be a little bit careful. Some things have similar names in this kit, so really make sure you know what you're doing. And I'm going to go ahead and add 100 microliters of this to each of my samples. So I've got my pipette, my tips. You may notice in this demonstration, I happen this moment to be using filter tips, and it's not necessary. It really depends upon what your requirements are. And again, it's more about protecting the sample rather than protecting me, but it's absolutely not required. In a classroom setting, we may just be trying to get a result. Of course, we want the result to be realistic and reasonable and representative, but this is not clinical forensics that we're doing here. So I'm gonna go ahead and attempt to add 100 microliters of this to each sample. I'll be making my own decisions each time I do this about whether I need to change tips each time. But actually what I'm going to do before I even go ahead and add that lysis additive is I'm going to remove the swab because it kind of just gets in the way and I don't need it. I happen to have some forceps, so I'm going to use those. Sometimes, well, I almost got one there. Sometimes the swab that's broken off inside sticks to the cap because there's actually a little hole inside. But if that's not the case, and if you need to get it out, if it's stuck, let's not confuse that one, you can go ahead and use a pipette tip. Oh, here's one where it's nicely stuck, so I'm just going to pull that one off. Sometimes it happens, sometimes it doesn't. But yeah, it just kind of gets in the way, so I like to remove it. You kind of want to be careful again that you're not exposing or touching the liquid in a way that you are causing cross-contamination between the samples. So use a little bit of judgment here and do whatever you think is safest for your particular workflow. Okay, so I will go ahead now and add 100 microliters of this to each one. I'm not going to touch any tips to anything, so I probably will get away with large volumes here and not have to change tips every time if I'm really careful not to contaminate that tip. You can do whatever you need. This particular preparation is going to ultimately use a column. Again, I find that it's just really works. We haven't tested a whole lot of different ways to prep this. I'm sure that there are others that might be a little bit less intense because you do have to plan for how much you can get done in a day. In my experience, because there's a lot of pipetting and a lot of coordinating and things like that between samples, there are places where you might actually want to divide this, especially if your class, you know, depending on what the length of your lab period is, you may not be able to get through all of this in one sitting. Or you might find that a different protocol works for you. That's shorter. Okay, so I have added this to all of them. I think I've capped them up. Now it recommends vortexing them. I do have a vortexer. I don't happen to be using one here. One of the things that you might notice when you're working with this is that the appearance of the solution becomes really pretty milky, a bit foamy. I don't think that's really inherent of any particular sample. It's just what's going to happen when the chemicals mix. But really in this step, as the name suggests, this is this lysis additive. So the goal here is to lyse microorganisms, which is in some ways not easy, right? There's many different types and oftentimes there's many different enzymatic treatments that you'll need to be successful with one or another. So obviously, or hopefully obviously, what you'll see in sequencing is only as good as your preparation here. So, you know, there may be things that you just couldn't lyse and didn't really get, even though they were present in the sample. So that's life. You have to do some more testing. All right, so I'm gonna make sure these are nice and capped up. Great. So the next step is to actually put this in the water bath at 65 degrees for 5 minutes. So I'm gonna go ahead and do that. All right, and then I'll set a 5-minute timer and I'll be back in 5 minutes. Okay, it's been 5 minutes and I have fished my samples out of the water bath. The cases now, you know, they are milky, foamy, however they look. And I've already labeled some tubes for my samples. I'm going to go ahead and transfer from these tubes into the microcentrifuge. And I believe I need to transfer a mil. So you'll see in the instructions here and elsewhere that sometimes it says a specific amount, but it says up to. So in some cases, you know, for example, if the sample is really dirty, which some of these samples might be, you have literally dirt, you might try to avoid pulling over the dirt, and it's more important to get a cleaner sample than rather get every single last drop and bring over something that is just going to contaminate and serve no purpose. But in my case, I think, see if I get my numbers somewhat in order here. I should be fine to get that mil. So I'm going to go ahead and transfer here. Of course, it's different samples, so I will have to transfer every time. Sometimes a little bit of soap bubble at the top, but I'll try not to get bubbles transferred. That's another thing that can happen too. I mean, I can see in my tube, I've got dirt. It's okay. I'm more actually worried about air bubbles, but in any case, 1 mil, and I'm going to do that for each sample. If there's a bubble at the top, you can always try to pop it just to not cover the pipette itself with soap foam. One of the other things that can cause you not to have that ml is I've noticed sometimes students spill things. So it's okay if you have a little bit less. If you were really, you know, spilled a lot of it, maybe there's still good sample in there. And you could use an empty sample tube that still has liquid to try to make up the difference, but you can make a decision. Okay, so I've got those 6 samples done. I'm going to save those. Not for any purposes other than to make sure I've got the labels all written down and documented elsewhere, but I don't believe that this is of any more use in terms of I can't go back and process it in any other way. So I'm just going to put it up to the side. And this is going to go into my centrifuge. We're going to spin it down for 2 minutes at maximum speed. In my case, it gives you an RPM, 14,000 RPM. All right, do that for 2 minutes. Okay, so it's been 2 minutes. And what you are going to notice when you take this out now, is that there's a little bit of a foam raft that is on the tubes. And what the purpose of pipetting, it may not be in every tube, it just depends on what was in the sample, but you'll see it pretty frequently. What your goal is going to be is to, if you have such a raft, not to suck that up. And then also at the bottom, sometimes there may actually be a little pellet of dirt, whatever else it is. So in the next step, we have the clean microfuge tubes, and we're going to transfer from our sample into the clean ones. And we don't want to get any of that material. We want to avoid it as much as possible. So the instructions are to transfer up to 700 microliters. Okay, so we're just going to go ahead and transfer carefully, avoiding any of that raft and any dirt at the bottom. So just be careful the way that you position your pipette. The raft actually usually can stick your pipette to get it underneath and then slowly suck up liquid. And I just managed to do it and get 700. I have a clean sample, leaving that raft behind. So I've got one tube that has a nice clean sample, and this one that's full of the dirt. We don't need that other tube, get rid of it. Okay, so let's do that to all of them. Some of them you may not have much of a raft. This one doesn't have much. There's something at the top, but not too much. But again, I try not to get it. Sometimes, like right now, it's sticking a bit to the outside of my pipette. So I'll be careful. And, you know, sometimes you can't help but get some of this in there. So do the best you can. The ones like this, it's hard because this raft kind of almost exploded. So that sample is a little bit dirtier. I'm not super worried about it. There's a little floaty stuff in it, but if you're ultra paranoid, you could spin it again, transfer again. I'm not going to go through that. Okay, so we've transferred that, and now we're going to add binding buffer, and that is also going to be 100 microliters. I'm just going to space things out because this is one of the ones that I can do this. And in this case, I need to use binding buffer 1. Be careful, there's more than one thing labeled binding buffer. So binding buffer 1, and I'm going to go ahead and add 100 microliters, and then I'm going to invert it and put on ice. So I've got set to 100, got all my tubes. I can be careful, the amount is large enough. I can do this without touching or splashing. And there'll be another chance, since we have centrifugation coming up, to get rid of things that have accumulated at the bottom of the tube. So the ones that are a little bit dirty, I'm not super worried. All right, and that's gonna sit there for 10 minutes. Okay, so it has been 10 minutes. I'm gonna take the samples off of the ice and put them in a centrifuge. This is also gonna be for 2 minutes at max speed. The sample has become really pretty cloudy sitting in the ice, so that's good. Likely here we've got some precipitation going on, so they look nice and cloudy. Some cloudier than others, but still cloudy. Okay, and we'll go ahead and spin that for 2 minutes. Okay, it has been 2 minutes. And you may or may not notice, I don't really see one here. A little bit of pelleting at the bottom. This one just has a slight scuff mark. But in this case, it's going to be easier to go ahead and remove the supernatant without actually getting any kind of pellet or anything that is going to contaminate or clog up downstream. So we can go ahead and transfer up to 700 microliters from this into these new tubes, which have also clean tubes. I've just labeled them while I was waiting. And pay attention to how much you actually carry over. It says up to 700. We're going to add ethanol in the next step, and the ethanol is supposed to be proportional. So 100 microliters per 100 microliters. So if you had 700, you're going to add 700. But if you weren't able to get that much, maybe you had some crud and you couldn't get that much, keep track of it. You can measure it by drawing up what you can in the pipette and then lowering the volume until you get a measurement. I've accidentally touched mine. And then you can measure it and then add a proportional amount of ethanol if you were unable. Maybe there was a lot of crud at the bottom of your sample. This one actually has not only a pellet, but it has like a little bit of a raft. This was that one that I had trouble before getting a clean sample, and now it's much, much cleaner. I think I've gotten rid of, haven't carried over any of that crud. I'm doing these 6 samples, and again, in a classroom environment, a student might be handling 1 or 2. Obviously, research project would be different. They could handle them all, but oftentimes we do this and we have, you know, a whole classroom of students. The kit that we're using for sequencing, the MinION kit, comes with 24 barcodes. It's to your advantage to actually use all 24 because otherwise you are kind of wasting a bit of the sequencing capacity. You're already a little bit overkill with even the smallest format that Nanopore offers. There are ways to scale it up. So you could decide if you want to process more samples and maybe do more flow cells, or there are different combinations, and we can discuss those later. Okay, I'm going to go ahead and add ethanol. So let me space things out here. And I need to add a proportional amount, so 700. And this is some 70% ethanol that I've freshly prepared. Probably get away with 1 pipette tip. There's going to be a number of steps involving ethanol and alcohols, and so tubes are labeled all on the top and the side. It's easy to get a drop somewhere and wipe off your label, so be careful of that. Also, the way that I pipette, I'm kind of realizing ethanol sticks to the sides of the tips sometimes, and there's little extra drips, so just be a bit mindful. It says here to vortex to mix. I'm not using a vortexer. Of course you could, but I'm just going to go ahead and invert them a couple times, and it's going to be enough to make sure it's thoroughly mixed. All right, so I have my ethanol in those samples. We've done all the precipitations to get rid of a whole bunch of proteins and dirt and everything else And so hopefully downward to something that's going to have really the nucleic acids. And that means it's time for the columns. So these are columns. In this kit, it says that we store them at 4C until we're using them. I mean, it's shipped at room temperature, so it's okay for a little while, but I guess this is new instruction. I've seen on them recently, so I guess it extends their life. We also need the collection tubes. So these collection tubes are probably 2 ml size, and that's because we have quite a bit of liquid, so we don't use a regular size 1.5 ml. I like labeling everything on the top and also on the side here, especially because it's going to get a fatter marker, easier for me to see. There's going to be, again, the chance that labels accidentally get wiped off. Also, because everything is separable, it's good to label it multiple places always, so you don't lose track. Okay, and I want to avoid touching or contaminating, so and as you handle them, you always of course want to avoid the inside of the tube, but also be mindful of the bottom, not to touch that. Avoid touching it as much as you can. Okay, let's label those. All right, so we're going to transfer some of the liquid in here, and as is pretty common, I think, with columns, usually you can't do it all at once, so we'll do it in stages, and it says to go ahead and do 700, so I guess it's just going to divide it 700 spin, 700 spin, so let's go ahead and do that. One of the things I'll draw attention to also here, let me space things out just so I avoid anything splashing in a way that is not conducive. The other thing is it's going to be a change in centrifuge speed, so just be prepared for that. Okay, so we're going to go ahead and apply 700, 700. This is such a large amount of volume. It's very easy to pipette. You don't have to worry about not making contact with the column. But in the future, sometimes it might be like the last steps where we have elutions, and you have to be a little bit careful to make sure the samples actually apply to the column, but that won't be the case here. Okay, I'm not going to show you every repeat, but we gonna go ahead and spin this down. We need to tune the centrifuge down to 10,000 RPM, and this is going to go for just a minute this time. Okay, it has been a minute, and we can remove those samples, and what you should notice is that all the liquid has gone to the bottom, and there's no liquid remaining in the top. I also like to have a napkin or two ready, so I can go ahead and pour that off into a waste receptacle. If for some reason the liquid was still remaining in the column, the instructions tell you to spin it down again. That could potentially happen if there was a lot of dirt or protein debris clogging everything up. If perhaps if the earlier pipetting was not as clean, the supernatants things are coming over, but that doesn't seem to be the case here. Oops, I accidentally took out a column and not the whole thing, so be careful that happens. Okay, it's okay. Just don't work too fast that you don't notice what's going on and accidentally touch it to the wrong place. Okay, So I'm going to space things out again, just so that in my mind I am clear on what's happening next and what I've done. And another 700. So you get what's coming next. This protocol, I don't want to use the word tedious bad way. It's a lot of pipetting, but we've done it with high school students pretty regularly, and they end up following the directions and getting good results. So it's routine. You still have to track what you're doing, but not too bad. Okay, so now what we're going to do is apply some binding buffer. Now this is a different binding buffer. We apply it directly to the column, so just be careful that you have the right one. I'm supposed to use binding buffer B, and we're going to actually use 500 microliters here. And it's such a large amount, it'll go right into the column, no problem. You can probably save pipette tips. So binding buffer B, 500 microliters. I am gonna still space things out. A little paranoid, no splashing. And then that's going to go ahead and spin down for a minute. Same 10,000 RPM. All right, that binding buffer just goes through and dab it. Okay, so we're again now going to wash. The DNA should be bound to that silica column, and that binding buffer, presumably by the name, helps it to stick. And now we're going to rinse away anything that's not DNA, hopefully. So literally wash, rinse, repeat. I'm going to do this twice. I'm going to apply wash solution A, flow it through, pour it off. Wash solution A, flow it through, pour it off. I'll either edit that out or you can fast forward. I've got to do it for real, but you get the idea. Okay, so we've completed the final applications of the wash buffer. And so now after, or the wash solution, now after we're going to do is going to spin it empty for 2 minutes, and that's to try to get every last drop of ethanol or alcohols out of the column. There'll still be a drop at the bottom, but we just want to make sure that the column itself is as dry as possible. And in the meantime, I'm going to prepare the elution tubes, just labeling them. They come with the kit. So this is 2 minutes at max speed. Okay. All right, so that has been spinning to dry it. Now, when I do this transfer, I want to make sure that I don't get any of that alcohol that's at the bottom. This is garbage. None of that can touch the column. We want to make sure everything is dry. So get rid of the flow-through collection tubes. Okay, great. Okay, so the next thing we're going to do is we need the elution solution. Okay, elution buffer B. So this is basically water and also probably some salts and buffers just to keep the pH right. And we're going to apply 50 microliters of this to the columns inside. Here I'm going to be careful because I really want that liquid not to get on the side of the column, but to really touch when I add it, that it really soaks into the white disc of silica that's there. So 50 microliters, and I should have tuned this down to 50 first. So I really need to look inside and make sure that I'm getting it directly on. If I was less careful, there's a chance that, because it's a small amount, it might, I don't want to touch the disc with my pipette tip or damage it, but I want to make sure that the liquid is not sticking on the side. And it's a fair amount of liquid. It's not, I've done things like this where you have like 10 microliters and you really gotta make sure it's there, but in this case, it's not a big deal. Just gotta do it carefully. Okay, and we're gonna go ahead and put these into the centrifuge. When we do, depending on the size of your rotor, how many you can fit in at once, may vary, because you have this open cap and then you have the inner side, so you have to kind of stagger things, but that's all right. Kind of just fits, I find, if I skip every other. And then pay attention too, because the amount of time, or rather the force that we're spinning this with is going to be changing. So we do one spin at 2000 RPM for a minute, and then we do another spin at maximum speed. So just make sure you consult the directions and get that correct. And this is basically the last step that we're going to do. Okay, this is the final product. There should be somewhere around 50 microliters at the bottom of that tube. The column gets disposed of, and this can go on ice because basically this is what we need for PCR. So it could, you could hold on to it, you could freeze it, because the next step is the PCR. The instructions say that you can do a second elution. I don't think that's really necessary, but you can decide that for yourself. And we're going to take these samples forward and do the 16S PCR, which is going to prepare us for sequencing.